Hi students, welcome to SPSVERSITY. This is Suresh, Senior Biology Faculty, having experience of 20 years. I taught biology for state level entrance examinations as well as national level entrance examinations like AAPMT. Now, I am teaching NEET biology. So I have a vast experience of 20 years in teaching biology for NEET. Today we are going to discuss about an important aspect in class 12, that is sexual reproduction in flowering plants. From this chapter, every year 4 to 5 questions come in NEET. So when you come to flowering plants, so in flowering plants, what is the purpose of flower? Flower is meant for only aesthetic value or decoration? No. The purpose of flower is sexual reproduction. So flower is meant for sexual reproduction in plants. So in flower, there are various parts like sepals, petals, stamen, and next corpus. So here in a flower, when you take the sepals and petals, they just help in reproduction. They are not directly involved in reproduction. So that's why they are called, that is, they are called non-essential parts of the flower. But whereas when you come to the male and female parts, so the male part is called androecium. The female part is called gynoecium. So which are directly involved in reproduction. That's why they are called essential parts of the flower. So here it doesn't mean that non-essential means they are not useful. They are useful, they just help in the process of sexual reproduction. They are not directly involved. Whereas when you come to androecium and gynoecium, they are directly involved in this. That's why they are called essential parts of the flower. So in a flower, if you take, it is floriculture means the cultivation of flowering plants. So in plants, before the formation of flower, so many changes will occur, that is hormonal changes and structural changes. So these hormonal and structural changes which lead to the development of floral primordium. What is meant by primordium? So primordium means it is an organ which is meant for subsequent development. That means it can develop into the different parts. So here floral primordium means where it develops into, floral primordium means it develops into a flower, that is, it develops into a flower. So in plants, there are a bunch of flowers. So we can see in most of the plants a bunch of flowers we can see. So these bunch of flowers which are present in plants, it is called inflorescence. So if you take inflorescence, the inflorescence axis, if you take the axis of inflorescence, so it is called peduncle. The axis of inflorescence is called peduncle. On peduncle, there are various parts, like small leaf-like parts. These are called bracts. And the angle between the bract and the peduncle, it is called axil of the bract. So in this axil of the bract, the floral bud arises. So floral bud means from which flower arises. So on an inflorescence, there are many floral buds in the axil of bracts, which will form a flower. So in a flower, as already we discussed, there are different parts. Like the male part is called androecium. The female part is called gynoecium. So this androecium and gynoecium will play an important role in the reproduction part. So first we are going to discuss about androecium. So androecium is an male part of a flower. It is called the male part of a flower. So what is androecium made up of? Androecium is made up of stamens. So again, androecium is made up of stamens. So these stamens, the number and their length will vary from flower to flower. So in all flowers, the stamens are not same in number as well as they are not in same in length. So here when we come to the structure of a stamen, so a stamen has a long slender part. What you are seeing, it is a stamen. It has a long slender part. So this long slender part of the stamen, it is called filament, or it is called stalk of the flower, stalk of the stamen. And above this there is a part that is called anther. That means a stamen has two parts. One is filament and another one is anther. So in filament, if you take in filament, filament is very long and slender. So the lower part is called the proximal end. It is called proximal end. The upper part is called distal end. So this part is called distal end of the filament. This part is called proximal end of the filament. So this proximal end of the filament attaches to thalamus, and distal end to this the anther is attached. So here this is anther. What you are seeing, this is anther. In this anther, it is made up of lobes. Again in anther, it is made up of subparts. They are called lobes. So in the diagram, there are two lobes. When two lobes are present, it is called bilobe. When only one lobe is present, it is called unilobe. In most of the flowering plants, there are two lobes, and very few plants there is only one lobe, that is called unilobe. In plants like hibiscus, we can see unilobe anther, where there is only one lobe. So here when you come to a bilobed anther, so anther is bilobed, which have two lobes. So this is one lobe, this is another lobe. And inside each lobe, if you take inside each lobe, if you go into the deep of inside of each lobe, inside each lobe there are again the spherical part. That is, in each lobe there are two parts. So these are called theca. That means in one lobe there are two theca. That means if it is bilobe, total how many theca will be there? 4 theca will be there. Okay. If it is unilobe, how many theca will be there? There will be 2 theca. See the difference. So there is a difference between bilobed and unilobe. In bilobed, only one lobe is present. That means only 2 theca are present. In bilobed, there are 2 lobes where 4 theca are present. In unilobed, there is only one lobe where 2 theca are present. So here we will come to the transverse section of anther. To study the structure of anther, we will go to the transverse section of an anther. So when you take the transverse section of a bilobed anther, it looks like this. So the anther is covered by, that is, if you take the outermost layers of anther, it is called anther wall. That is, anther is well protected by anther wall. And below this anther wall, there is an fertile tissue that is called sporogenous tissue. So sporogenous tissue, why it is called fertile? Because from this sporogenous tissue, pollen are produced. The anther wall will give protection as well as the breaking of anther. So if you take an transverse section of an anther, it has anther wall and sporogenous tissue. So the anther wall, if you take the function of anther wall, it is to give protection as well as the dehiscence. Dehiscence means the breaking of anther to release pollen grains. The function of sporogenous tissue is to form pollen. It will form pollen grains which will have male gametes inside. So when I come to anther wall, so when you take the anther wall, so the anther wall is made up of 4 layers. So when you start from outside, it is made up of epidermis, endothecium, middle layers, and the innermost layer is epithelium. So anther wall has 4 layers. Outermost layer is called epidermis. The second layer is called endothecium. And the third layer is called middle layer. And the last, which is the innermost layer, is called epithelium. Each and every layer, if you take each and every layer of this one, the anther wall, it will have each and every have specific function. So here this, when you take the 4 layers, when you take the 4 layers, In the 4 layers of anther wall, the outer 3 wall layers means epidermis, endothecium, middle layers. Their function is giving protection. And dehiscence means breaking of anther so that the pollen grains are released. So the 3 layers function is protection and dehiscence. And there is an innermost layer, that is tapetum. So tapetum is having an unique function. Besides it is giving protection and dehiscence, it plays an important role in nourishment. Nourishment means giving food to developing pollen grains. So to give nourishment, the cells of tapetum has dense cytoplasm which is rich in food materials and have more than one nucleus. Generally in most of the cells there will be only one nucleus, but here they will have more than one nucleus. How it is possible? Okay. Why more than one nucleus is present? The reason for this is during the cell division process, only nucleus will divide, whereas the cytoplasm doesn't divide, which leads to the multinucleate condition. Okay. As cytoplasm doesn't divide, there are many nuclei in the cells of tapetum. Okay. So here in this cytoplasm, as cytoplasm doesn't divide, there are more than one nucleus in the cells of tapetum. Here when you come to this, the next part of the anther, that is the sporogenous tissue. So the next part of sporogenous tissue is, the next part of anther is sporogenous tissue. So at each corner there is a sporogenous tissue, so which is called microsporangium. So what does this sporogenous tissue do? So this sporogenous tissue will form the microspore mother cells. It will form the microspore mother cells, which are diploid. They have 2 sets of chromosomes. So this microspore mother cells, each microspore mother cells will undergo meiosis. On meiosis, from each microspore mother cell, 4 microspores are formed, where each and every one is haploid. Okay. Due to meiosis, the diploid is made into haploid. And these 4 microspores which are formed, they are attached with each other. So this entire structure where 4 microspores are present, it is called tetrad of microspores. So this tetrad of microspores on dehydration. Dehydration means loss of water. They get separated. Then each one is called pollen grain. That means from one microspore mother cell, 4 pollen grains are formed. So from one microspore mother cell, 4 pollen grains are formed. So here you can see the process where from microspore mother cell to microspores are formed. So this process is called microsporogenesis. So here from the cells of sporogenous tissue, microspore mother cells are formed, which will undergo meiosis. So here 4 microspores are formed, which are haploid. As 4 are attached, it is called tetrad. On dehydration, the microspores separate and they form pollen grains. So here, from one microspore mother cell, 4 pollen grains are formed. So from one microspore mother cell, 4 pollen grains are formed. You can write the same thing like 1 tetrad of microspore is formed. Okay. So as meiosis takes place in microspore mother cell, 4 pollen grains are formed. Tetrad of microspore means 4 microspores are formed. So there is a difference between microspore and pollen grains. Microspores means they are stay together, 4 microspores will stay together, whereas when they get separated, they are called pollen grains. And this process, that is formation of microspores from microspore mother cell, it is called microsporogenesis. So this is about microsporogenesis. So by the end, we got pollen grains. So which represent, the pollen grains are called the first male gametophyte. Okay. Here gametophyte means the haploid structures. So here you are seeing various types of pollen grains which are in different shape. Okay. So this pollen grains which form the first male gametophyte, they carry male gametes and play an important role in sexual reproduction. So when you come to the structure of a pollen grain, when you take the structure of a pollen grain, the pollen grain is covered by 2 wall layers. That is, the outermost wall layer is called exine and the innermost one is called intine. It has 2 layers, that is exine and intine. Exine is very hard. It is tolerant to strong chemicals and alkalis, even high temperatures. And it is made up of a chemical called sporopollen. It is made up of a chemical called sporopollen. And on exine, there are gaps on exine. Gaps means where there is no sporopollen. So these gaps, they are called germ pore. This gap is called germ pore. What is the purpose of germ pore? The purpose of germ pore is to emerge the pollen tube. During the germination of pollen grain, pollen tube emerges from germ pore. And when you come to inner wall, the inner wall is called intine, which is made up of pectocellulose, which is very thin and it is made up of pectocellulose. It is a continuous one. It doesn't have any gaps like exine. It doesn't have any gaps like exine. So pollen grain, the wall of pollen grain is made up of exine and intine. outermost is called exine and innermost is called intine. So here you can see this is exine, this is intine. And above this there is a pollen kit layer where the pollen grains are carried by insects. In such cases, it is a layer called pollen kit for attachment to insects. Okay. So here when you take the pollen grain, okay, so the pollen grain will be in different size, shape and design. What you are seeing the diagram, there are different size and shapes. Okay. Depending upon the species, they are in different size and shapes. So these pollen grains are very resistant to strong chemicals due to presence of sporopollen. So that's why due to presence of sporopollen, pollen grains are preserved as fossils. In the process of evolution, pollen grains are preserved as fossils. This is done which is natural. So when we come to artificial means of preservation for our human benefits, so pollen grains are preserved in -1926 degrees in liquid nitrogen, where they are preserved at very low temperature. That's why it is called cryopreservation. And when you come to this, pollen grains, they are rich in food. That's why they are used as a very good source of nutritional values. That's why they are used as a food supplements or nutrient supplements of athletes and who are race horses. So these are a few benefits of pollen. They have good nutritional value. As they have good nutritional value, they are used by athletes and race horses as a food supplements. At the same time, there are some harmful effects of pollen grains. Not all the pollen grains, a few pollen grains. Like pollen grains, some few pollen grains cause allergy, bronchial afflictions, asthma, bronchitis. Like pollen grain of Parthenium, which is called carrot grass. Parthenium is commonly called carrot grass, where they cause asthma and bronchitis. Okay. So when I come to lifespan of pollen grains, it depends on temperature and humidity. So there are pollen grains where lifespan is in few minutes as well as in months. Like if you take rice, the pollen grain lifespan is few minutes. Within few minutes, the lifespan ends. But whereas when you come to the families like Solanaceae, Rosaceae and Legumiaceae, they have months of lifespan. So that's why they're stored in, the pollen grains are stored in pollen banks at very low temperature, artificially at 196 degrees in liquid nitrogen. So where they can be used in crop breeding programs. They are used in crop breeding programs. So now we are going into the deep parts of pollen grain. So till now what we discussed is the walls of pollen grain, that is exine, and the next one is intine. When I go into inside of pollen grain, there is a nucleus which is haploid and there are two vectors. So this nucleus which is present inside the pollen grain, so this nucleus undergoes a mitotic division, that is a cell cycle. So due to mitosis, two nuclei are formed. The two cells are formed inside the pollen grain. As this mitosis is an unequal mitosis, which leads to one large cell and one small cell. So the large cell is called vegetative cell and the small cell is called generative cell. That means at this condition, there are two cells in the pollen grain. The larger one is called vegetative cell and the smaller one is called generative cell. Okay. So there are two cells, one is vegetative cell and another one is generative cell. The vegetative cell is large, which is have abundant food reserves, and nucleus is irregular shape. Whereas when you come to generative cell, it is small, it has a nucleus, it floats in the cytoplasm of vegetative cell. So when the pollen grain is at this stage, that means when it is in two cell stage means having one vegetative cell and one generative cell. So vegetative and generative cells, they are, when they are present, it is called two cell stage. At this stage, the pollen grains come out of anther in most of the plants. That is, in 60% of angiosperms, they will come out. In remaining plants, what happens? In remaining plants, before they are released from anther, the generative cell will divide and form two male gametes. It is from generative cell, two male gametes are formed, where two male gametes are formed from generative cell. That means in few plants, that is in remaining plants, in a pollen grain there is one vegetative cell and two male gametes. That means total how many cells here? 3 cells. So total it is 3 cell stage. So in a pollen grain, that is in most of the plants, when the pollen grains are at 2 cell stage, they are released. In remaining plants, that is in the remaining plant, they come out of 3 cell stage. So 3 cell stage means one vegetative cell and 2 male gametes. So this is what happens inside the pollen grain. And already we discussed in vegetative cell there is a food reserves. It is abundant in food reserves. As it is abundant in food reserves, they are used as nutrient supplements. Like you can see, there are pollen tablets where they are rich in nutrients. Bee pollen, like bee pollen cream. There are different energy shake which are made from this pollen grain, which are rich in nutrients. So here you can see there is a development of pollen grain takes place. So here the development of pollen grain means we have, there is the development of pollen grain takes place partially inside the anther as well as partially after coming out of anther. So for example, if you take, in most of the plants, that is in 60% of the plants, the pollen grains are shed at 2 cell stage. 2 cell stage means when the pollen grains are having one large cell, that is one vegetative cell and one generative cell. So here in such type of plants where the pollen grains are shed at 2 cell stage, that is for formation of vegetative and generative cell, one mitosis is required. Now they have come out of anther. Where they will reach? They will fall on stigma. After reaching stigma, when they enter into ovary ovule, again the generative cell will divide by mitosis and forms two male gametes. The male gametes are formed from the generative cell. That means in such type of plants where the pollen grains are shed at two-cell stage, we can see the development of pollen grain takes place partially inside the anther as well as partially outside the anther. So this is called the development of the male gametophyte. When you take the three-cell stage pollen grain, in three-cell stage pollen grain, so complete development takes place when it is inside the anther only. Inside the anther only, the two mitosis divisions takes place, which will lead to formation of two male gametes and already existing one vegetative cell. So here, this is how the development of male gametophyte takes place. So now, there are two male gametes inside. There are two male gametes inside the pollen grain. So pollen grain is ready for the next process of sexual reproduction, that is fertilization. Okay. So whatever the pollen grains are ready for the next stage of fertilization. So next they are ready for the next stage of fertilization means they are ready with the male gametes. Okay. From where these male gametes are formed? So these male gametes are formed from the generative cell. The male gametes are formed from generative cell. So the function of vegetative cell is nutrition. That is, the function of vegetative cell is nourishment for generative cell. Okay. So that the pollen grains are formed from the generative cell. So this is how we can see the development of pollen grains. Okay. So here when you come to the development of pollen grains, so here you should remember some numerical things, like during the formation of the pollen grains from one microspore mother cell. So from one microspore mother cell, 4 pollen grains are formed. The microspore mother cell is diploid. Pollen grains are haploid. So here you should remember the ploidy and number of pollen grains are formed, and as well as the number of chromosomes. Suppose if microspore mother cell is having 24 chromosomes, how many chromosomes will be there in the pollen grains? Each pollen grain. Obviously, it is half of it. It is nothing but 12 chromosomes in each pollen grain. The same thing applies to microspore. They will have only 12 chromosomes in microspore. So if you remember the ploidy, you can write the chromosome numbers, whatever the chromosome numbers are given. And at the same time, we should remember the number of pollen grains formed from the microspore mother cell. Suppose if there are 10 microspore mother cells, how many pollen grains are formed? So from each mother cell, 4 are formed. That means 10 multiplied with 4 will get 40 pollen grains. So such type of numericals you should remember for answering questions in the NEET. Okay, from 10 microspore mother cells, 40 pollen grains are required. So for formation of 40 pollen grains, how many meiotic divisions are required? That means each microspore mother cell will undergo one meiosis. That is 10 microspore mother cells, each one meiosis. That means how many 10 meiotic divisions are required. So here you should remember the meiotic divisions, the number of pollen grains, the ploidy levels. So when you remember these number of chromosomes, the ploidy, the pollen grains, that is from one microspore, 4 pollen grains are formed on one meiosis. At the same time, during the development of pollen grain, so already we discussed during the development of pollen grain, in the development of pollen grain, from starting from haploid nucleus to the male gametes, total there are 2 mitotic divisions takes place inside the pollen grain. On 2 mitotic divisions, that is, on first mitotic division, what are formed? One vegetative cell and one generative cell is formed. And second mitotic division, only generative cell will undergo second mitotic division and it will form 2 male gametes. That means on 2 mitosis we will get by the end 2 male gametes. So here each pollen grain has 2 male gametes. Each pollen grain has 2 male gametes. And they are formed by 2 mitotic divisions. Suppose if there are 10 pollen grains, how many male gametes are there? Means 10 multiplied with 2, 20 male gametes. And in each pollen grain, 2 mitotic divisions takes place. So here there is a chance of getting numerical questions while answering the questions. So numerical questions means you should remember the numbers, the ploidy, the number of meiotic divisions, the number of mitotic divisions which occur during the formation of pollen grain as well as during the formation of male gametes. So, and the next part you should remember is the 2 cell stage and 3 cell stage. 2 cell stage means what are the cells present? One vegetative cell and one generative cell. 3 cell stage means one vegetative cell and 2 male gametes are present. So what are the layers present on in a pollen grain? That is exine and intine. So exine is very hot, which is made up of sporopollen. Okay. So these are the numericals you should remember in the part of the androecium part. You should remember these are the numericals which you should remember. And at the same time, here you should remember the parts of the anther wall in a sequence. That is, if you start from outer to inner, epidermis, endothelium, middle layers, tapetum, in sequence. So such type of questions will come like sequential questions, where they ask, write the parts in a sequence from outside to inside or from inside to outside. Inside to outside means tapetum, middle layer, endothelium, epidermis. Okay. So like this, and at the same time the functions of these 4 layers. As we discussed already, the anther, the 4 layers, that is outer 3 layers are having the function, that is their function is the production and release of pollen. The tapetum function is nourishment. And tapetum is having a specific feature, that is the cells of tapetum are rich in cytoplasm and have more than one nucleus. The reason for this is nucleus will divide, cytoplasm doesn't divide. That's why it leads to the multinucleate condition. And below the tapetum, what is present? Sporogenous tissue. So sporogenous tissue is haploid. So here you should remember the structures which are haploid, the structures which are diploid. Microspores, pollen grains are haploid. Microspore mother cell, sporogenous tissue, remaining whatever you come across, that is the epidermal cells, the endothelium, middle layers, they all are diploid. Before meiosis, whatever the structures are present, they are diploid. After meiosis, whatever the structures will come, they are haploid. The vegetative cell is haploid, generative cell is haploid, the male gamete is haploid. Pollen grains, microspores, vegetative cell, generative cell, male gamete, these are haploid structures. When you remember the haploid and diploid structures, you can easily answer the chromosome numbers, if any particular chromosomes are given. So in plants, if we take specifically, most of the cells are haploid except gametes. So except gametes, remaining all except gametes, pollen grains, the male gametophyte, female gametophyte, remaining all are diploid. There is only a diploid structure in plants, that is endosperm. So if you can remember these ploidy of the structures, you can answer the structures, that is the questions like when they ask regarding about giving an example of chromosome number. Suppose if you take a leaf cell, in a leaf cell the chromosome number is 16. For example, the chromosome number in a leaf cell is 16. So leaf cell is diploid. Then you can answer 2n is equal to 60. Whatever it is asked, like what is the chromosome number in pollen? Pollen is haploid. That means half of this 8. Microspore is haploid. Half of this 8. So like this, you can answer when you remember the ploidy of structures. And the next thing, the difference between the pollen grain and microspore. Microspores are in combined state where 4 are present, whereas the pollen grains are in separated by dehydration. Okay. So here, what is meant by microsporogenesis? That is formation of microspores from microspore mother cell. So these are the terms you should remember in the androecium part, that is in the structure of stamen, in the development of pollen, and in the structure of pollen grain, in the development of pollen grain, and the uses of pollen grain, as they are used as food supplements, as they are rich in food. In the vegetative cell, it is rich in food, it is rich in supplements. So this is about the stamen, that is the female part androecium. It's the female part, the male part androecium, the stamens, and the development of the pollen grain, how pollen grains are formed, and what is the uses of pollen grain for humans, as well as how they can be stored, and the next aspect is how they can be stored naturally as well as artificially. So thank you very much. We'll meet in the next session, continuing the topic of gynoecium. Thank you all.